I think it's important to start off by understanding who the enemy is. I think as oncologists, even as we're developing more and more effective therapies, I think we've come to realize that really a central obstacle to curative cancer care is the idea that a resistance can develop, that this is generated by tumor evolution. And fundamentally fueling this process is the fact that cancers have intratumoral heterogeneity. Not a new concept. It's been known for quite some time, ever since the 70s, when we realized that somatic mutation was part and parcel of what a cancer cell is, and that this would generate obstacles to therapy with the heterogeneity. But I think really what's changed is that we now have much better ways to measure it, understand it, monitor it. That's really been born out of the progress made with availability of next generation sequencing. And so I've looked at this question through the lens of CLL. So this is the disease that my group studies. These kind of themes are broad across cancers, but we've focused on chronic lymphocytic leukemia, which, just as we'll all recall, is a chronic malignancy, which makes it really actually quite ideal for studying tumor evolution, because in the same patient with this indolent disease, we can sample multiple times and actually watch tumor evolution in action. I'll just remind you that there is quite a bit of clinical heterogeneity in this disease. Most patients go through watch and wait initially, but like other cancers, there's acceleration in the pace of the disease and ultimately progression. Combination chemoimmunotherapy is still the standard first-line therapy, but there's been great excitement and rapid adoption of targeted pathway inhibitors. And so I just want to say that like other CLL investigators, there's been a long interest in trying to understand what is the basis of this clinical variability that we see in patients. There's always been a search for markers that can distinguish between patients who do well and others who succumb to their disease rapidly. But I would say that really there's been a tremendous amount of progress since 2010 with availability of next generation sequencing that has allowed us to really truly understand the genetic alterations underlying this disease. We're understanding ever more the transcriptional alterations and now epigenetic alterations as well. And there's been, as in other cancers, many fruits of next generation studies. Our group has kind of led the charge in kind of defining the mutational landscape of these tumors. I'll show that in the next slide. We've identified mutations that can affect cellular pathways in CLL. We've identified and been able to characterize the intratumoral genetic heterogeneity that is a common feature of CLL that has allowed us to understand what are the alterations that happen earlier versus later in disease, and to understand some of the evolutionary trajectories. And this is just a visual of kind of our latest version of this work, in which we profiled more than 500 cases of CLL. This is tumor and matched samples. Across the left side is all the different significant driver alterations that we identified. And I think just a bird's eye view just tells you really there's really quite a variability across these more than 500 patients. There is not one single uniform profile for all the patients, really demonstrating the independent evolutionary trajectories that are undertaken. One of the key features that we've identified across our studies is that, again, I mentioned the intratumoral heterogeneity, and for the most part, what we've been identifying is that there are small populations that are already existing within the tumor population that, under the selective pressure of therapy with cytoreductive therapy, that there is a shift in the genetic profile, and that what is originally a minor population can become the major population later on that can provide resistance to treatment. And this kind of concept of subclones that can portend future relapse, and that is a marker of ongoing active evolution, we tested in a large cohort of CLL samples. So initially we looked at those a group of 149 cases, and those cases that had subclonal alterations in driver mutations, again, suggesting active evolution, had an earlier need for treatment, an earlier need for retreatment. And then we went on and looked at a dedicated treatment cohort, again, from a phase 3 clinical trial, and we saw that this kind of concept held regardless of whether they received FC versus FCR chemotherapy. And more recently, we've been looking at targeted inhibitor therapy. So this is ibrutinib, which has kind of taken the field by the storm. And I showed this example of a patient who was originally treated with chemo, then got ibrutinib, relapsed after 3 years, and we sampled this patient at subsequent times across this more than 7-year period. And when we perform genetic profiling, this is using whole-exome sequencing, where we're identifying all the different mutations, we're performing cluster analysis, we're identifying what the different subclones are. What the colors tell you is that there's really different genetic eras in that patient in relationship to the therapy, so that while originally there is a number of alterations that are common throughout all the different subclones, you can see in the green that this was what was the composition prior to chemotherapy, that by the time 3 years later the patient is receiving targeted inhibitor therapy, there is the blue clusters, and there is a bad subclone in there, cluster 4, that has a lot of bad stuff in there, and this ends up being the parent that then subsequently generates the 4 different progeny that have mutations, not in 1, but 4 different mutations that allow the tumor to overcome the B-cell receptor signaling inhibition. So I say this just to say that I think this is a problem that we face, that the capacity, the more that we study this, we realize the capacity of resistance is already inherent within the tumor, that there is so much heterogeneity in there. And so at least in CLL, what we see is that in the absence of therapy, there is already these different subpopulations depending on what therapy you give that patient, whether it's chemotherapy or targeted therapy A or B. There's going to be a push and pull, and that there'll be emergence of different subpopulations depending on what that selective pressure is. And so with this kind of very long-winded preamble, I just want to kind of set the stage and ask the question, given that each tumor arises from independent evolutionary events, what is our best strategy for eradicating that tumor? And what I would say is that as much as next-generation sequencing has provided us with so much wealth of information about the driving genetic events in each tumor, it also presents us with an opportunity, because for the first time, we're really able to systematically identify mutations that are specific to the tumor, realizing that some of those mutations will cause amino acid changes that, when translated into peptides that are proteolytically cleaved within the cell and have the potential to bind to HLA molecules, they can traffic to the surface and be displayed where they can be recognized by T cells. And in particular, what happens with the presence of the DNA mutation, they're causing new changes, so we call those neoantigens. And those neoantigens may be quite discrete, so generated by a single amino acid, or they can be longer stretches of amino acid change. So this is what we call a neo-orf, a new open reading frame. So for example, if there is a splice site mutation, for example, there could be a retained intron. If there is a insertion or deletion, this could cause a frame shift. If there's a read-through mutation, that is also new sequences that are specific to the tumor, not present in the germline. And finally, if you have a gene fusion, for example, whether it's causing a in-frame or, more interestingly, an out-of-frame sort of joining, that can also generate new sequences as well. And so I would put forth that as much as we're worried and concerned that there's all this kind of heterogeneity, not only amongst individuals, but within individual cancers themselves, I would pose that raising a standing army of T cells, this kind of adaptive immune response, really provides us with the potential to address the diversity of antigens that are present and unique to each tumor and each patient. Why do we think neoantigens are feasible and an important class of antigen to target? Well, it turns out that there's a long-standing history of thinking about neoantigens, and I point out just a number of studies dating back to the 90s, both in animal models where targeting even single neoantigens was effective for eliminating tumor, to very elegant human studies looking at long-term survivors, unexpected long-term survivors in melanoma, characterizing the antigen profile and realizing that a lot of the durable responses were related, enduring responses were related to neoantigens, not the shared GP100s or melanase or tyrosinases. There's a long series of studies in follicular lymphoma, vaccinating against patient-specific mutated immunoglobulins. And if you scour the literature, really there's kind of, across all the different cancers, there's been anecdotal studies where mutated antigens have been noted to be quite immunogenic. But really what was lacking was any sort of way to systematically find these neoantigens. It's just too daunting on a patient-by-patient level. And in my postdoc days, I remember doing T-cell cloning, and it's not for the faint-hearted. So I think the other thing that we're helped with, aside from the availability of next-generation sequencing, is the maturation of predictive algorithms of HLA binding. So when I was a postdoc, all we could do was predict HLA-A2, which is a great start, but it's really quite limiting. And what's really matured over the years is kind of the development of these databases that allow us to train by machine learning and using neural nets to generate predictive algorithms across the HLA alleles. So if you can imagine that you have a mutated epitope compared to germline and you tile across in the 8, 9, 10 mers that we know are the size of class 1 peptides, and then we ask computer, these are the alleles that we're interested in, and can you spit out for us what are the predicted binding scores? What you see here is numbers greater than 500 are considered non-binding. If it's less than 150, it's intermediate binding, less than 50, it's strong binding. So in this matrix, you'll see 2 peptides that perhaps would be worthwhile kind of considering for testing further. And in my own group, we've kind of looked at this a little bit more deeply. We've synthesized hundreds of peptides that were predicted, and working together with Alex Sete and LIAI, we perform competitive binding assays against reference HLA alleles and peptides of known binding scores. And indeed, we see that if you remain within the less than 150 to 200 IC50 nanomolar score, it actually is consistent with good experimental binding. So kind of with this kind of idea in mind, early on, we invested very deeply in trying to generate a neoantigen discovery workflow that starts with the idea of taking tumor specimens, performing DNA and RNA sequencing, having HLA typing information available, performing mutation calling, but integrating the HLA typing information so we can perform prediction of binding epitopes. And then we wanted to ask the question, if we could predict these epitopes, do they in fact generate T-cell responses? And so thinking, where can we get samples that have HLA typing information? Well, we did have samples available from patients who had gone through allogeneic stem cell transplant. And at the time, we had a study going on in CLL patients who were vaccinated with whole tumor cell vaccine following allogeneic stem cell transplant. And we had a number of patients who had enduring responses. So we asked the question, in those patients with long-lived responses, can we ask if those T cells in those patients actually recognize predicted personal neoepitopes on the original tumor? So we went forth and we sequenced the tumor. We were able to identify the mutations. We could predict the peptides based on that patient's HLA type. We generated those peptides, and then we tested those T cells by pooling the peptides and asked whether there was reactivity. And what you can see with those spots is that, in fact, there was interferon gamma secretion consistent with recognition against one pool and not the other 2 pools, and not against the control peptide. And upon deconvolution of that pool 1, we could see that the T cells from this patient were reactive against mutated FNDC3B, which was a predicted peptide from this patient's tumor, not against the corresponding wild type. And those same T cells could then recognize the patient's own autologous tumor. And then Marcella talked a bit about persistence. Certainly when we track the V-beta sequence of these mutant epitope-specific T cells, we could find enduring evidence of T cells reactive against this neoepitope circulating in the blood of this patient even 30+ months following stem cell transplant at the time that the patient was in persistent MRD negative disease. Now that was a situation where we had HLA typing information because the patient had gone through stem cell transplant. At that point, we really wanted to be able to go on and access information from all the legacy exomes that are in TCGA. And it should be that with exome sequencing that we should be able to get HLA typing information from those exomes. It turns out that that is actually not the case. If you consider that when we do alignments for the sequencing data, we are basically taking the tumor sequence, and in the reference sequence there is only one HLA-A sequence, but consider that there are several thousands of HLA alleles, so you can already see the magnitude of the problem, that there's really very great difficulty using these short reads to align to these highly polymorphic loci. So we actually had to do some tool development. So this is only to say that we expended quite a bit of time trying to develop a strategy that actually works very well, in which we created a secondary library and we took into account all the different HLA-A and B alleles, the tens of thousands of alleles that are in there, so that we can do exquisite alignment. And now we can create a secondary inference step where we kind of take into account ethnicity, as well as the fact that HLA alleles sometimes are homozygous rather than heterozygous, so that created another challenge. But suffice it to say that our tool polysolver, which is in the black bars, when we tested it against HapMap samples for which there was already existing exome sequencing data, existing gold standard HLA typing data, that our algorithm actually performed exceedingly well. So now we can go back to TCGA, where there's tens of thousands of tumor samples. We know very well from our friends at the Broad Institute, where they've measured the somatic mutation frequency across these tumors. We know what they are. We also know what the missense mutations are. And then we could apply polysolver, get the HLA typing information, and ask the question, what is the neoantigen load? And then we see in these tumors that the pattern and the load of neoantigens is quite similar to what we see in terms of the missense mutations, because that's the bulk of the type of alterations from which our predictions are generated. So I think what you can see from here is that melanoma leads the pack in terms of somatic mutation rate. It is also the tumor that is predicted to have the highest number of neoantigens, and it kind of tapers down as we go. Now, this type of approach has been taken by other investigators as well, and by now there's quite a number of elegant studies that have been published asking the question, is mutational load or neoantigen load in fact related to response to immunologic therapy? And I point to a number of publications by colleagues in Memorial who have looked at response to ipilimumab and see that mutational load is associated with response there. Other colleagues in New York have looked at lung cancer, and again, response to PD-1 therapy also seems to be associated with mutational load, and the same for colorectal cancer. We took it one step further. We asked, well, if neoantigens are so immunogenic, shouldn't this also be associated with presence of a cellular infiltrate that would come in and want to recognize tumor cells that are expressing these antigens? So joining together with our colleagues in the GU oncology group, Sachet, who had developed, in my group, who had developed polysolver, he worked together with Brooke Howitt to look at high mutation rate GYN tumors compared to lower mutation rate, and in fact sees differences in predicted neoantigen load. And it is those tumors that have higher mutation rate that also do have a higher infiltrate of CD3, CD8 positive T cells. We had the opportunity to look even more directly. This was a really fun partnership that we worked together with Levi Garraway's group. So this is Marios Giannakis, a really talented young oncologist who worked together with Charlie Fuchs and Suji Ogino to kind of collect a series of more than 600 tumor normal pairs from patients with colorectal cancer that were collected on the Nurses' Health Study as well as the Health Professional Study. These were FFP specimens for which we could do whole exome sequencing and really look at all the different genomic attributes. There's more than 20-year follow-up. Shuji Ojino had already characterized the kind of the infiltrate pattern, so the cellular infiltrate pattern of these specimens. So it was really this wonderful opportunity to marry together genomics, clinical data, and also pathology information. And what we saw when we did that was we noticed that with increasing intensity of infiltrate, in fact, this was associated with a higher number of neoantigens, and that the nature of the infiltrate was that it was predominantly CD45RO positive, consistent with T cells that have previously experienced antigen. So I think this was kind of a nice kind of confirmation of these concepts of in a kind of a natural cancer history that neoantigens are immunogenic. And so what I've shown you so far is that there's been a lot of various studies examining neoantigen load related to clinical response. I've described to you in our own transplant experience that neoantigen reactive T cells can be expanded in the setting of effective clinical response. Steve Rosenberg has also looked at this in tumor infiltrating lymphocytes, very wonderful data. Various groups have looked at checkpoint blockade and also see that these neoantigen reactive T cells seem to be at the heart of these effective responses. There's also been exciting data to show that neoantigen-specific T cells can kill in vivo, both in patients and in animal models. And so another question that we wanted to also ask is whether neoantigens participate in this kind of crosstalk between tumor and the host and whether they evolve together. And for that, I want to remind you that having generated polysolver, we had the ability to type germline sequences, and this then gave us the opportunity across tumors to then compare against the germline whether or not there was an actual point mutation in HLA. And so this ended up being kind of a HLA mutation detection pipeline that worked quite efficiently. And when Sachet looked at close to 4,000 samples across the different tumor types, he noted that, in fact, there were quite a few HLA mutations. Across the top, it shows that a lot of them, they're kind of scattered throughout the gene locus, and that many of them are out of frame or nonsense, suggesting that they're loss of function. And in particular, colorectal cancer ended up being significant as a tumor that seems to have a lot of mutations in HLA. So with this kind of information, we asked, well, if HLA mutations are there, maybe a hypothesis would be that HLA mutations are more likely to rise in tumors infiltrated with CTLs and would provide strong selective advantage to emergent subclones. And so working together with Mike Rooney, at that time a really talented computational graduate student, we had matched RNA sequencing data from a series of tumors and asked, is there any signatures that would be suggestive of this? And it turns out that in an unsupervised analysis that the top hits were all related to, in fact, CTL T-cell function. And that's shown here, where really we see evidence of cytolytic activity. So these are tumors that are what's more enriched in tumors that have HLA mutations compared to not, is those markers associated with cytolytic T-cell function. And so the kind of the emerging picture, and when we went back and looked at Marius Giannakis' data, in fact, those colorectal cancers that had an increasing number of TILs were also the ones that had an increasing number of HLA mutations, and that was irregardless of whether the overall mutation rate. And so the take-home that I want to convey to you, and this is kind of our working model of what's going on, this is kind of the thought behind a lot of the ongoing work in my group as we kind of begin to look at different patient cohorts, is that if neoantigens are truly as immunogenic as we say, and there's several lines of evidence to show that, then one might imagine that they certainly can provoke a host immune response. If the host immune response is actually effective, it will lead to elimination of the tumor. But with that type of selective pressure, it does provide impetus for escape, because those subclones that have mechanisms that kind of can overcome that selective pressure, whether it's through affecting the antigen-presenting machinery or immune regulation, can then lead to progression of the tumor. And I think we're already beginning to see examples of that in various publications that have come out in the past year in the setting of checkpoint blockade. So at this point, I want to shift to asking the question, why vaccines? And I think Bruce asked this question already. And if we want to go with vaccines, how can we build more effective cancer vaccines? And I really, at this point, Marcella alluded to how the landscape for therapy has really shifted over the last 5 years. And I think many of us will remember when these first two papers came out, it really made heads turn, especially in solid tumor oncology, that the kind of the promise of immunotherapy. But I will point out that with checkpoint blockade therapy, certainly it's been immensely effective across different cancers. In the blood malignancies, we know that Hodgkin's lymphoma, it is highly effective, and there's been amazing responses in melanoma and lung. But we also know that there's suboptimal responses in many settings, and I point to the example of myeloma, where in a study really is absence of any sort of response. Now, one approach is to combine the antibodies, but I would say that in the example shown here, these are figures taken from a study published last year where more than 900 melanoma patients were treated with a combination of anti-PD-1 and anti-CTLA-4 blocking antibody. And as you can see from the yellow waterfall plot, there are more responses compared to either single agent alone, but it was at the cost of increasing toxicity, where 55% of patients had grade 3/4 toxicities. And so I think the question that we have to ask is, how can we increase efficacy while minimizing toxicity? And one way to think about it is, can we kind of decouple the steps? Can we somehow, again, a concept that Bruce was alluding to, is can we somehow sensitize in an anti-tumor fashion first and then overcome immunosuppression? And for example, one might imagine a vaccination approach or a cellular, cell-based approach followed by checkpoint blockade. And what could a vaccine do? Well, it could increase T-cell infiltration. It could recruit new T-cells. It could increase the repertoire of tumor-specific T-cells. And again, I think our goal here is to increase efficacy, minimize toxicity. And the way that I've kind of been thinking about it is that a cancer vaccine certainly has the ability or the promise for steering the immune response and effectively, given that we now have ways to effectively release the brakes, we have good adjuvants that can kind of really put some power into the response and effectively get at the tumor. So cancer vaccine. So what do we think at the minimum we need to address if we want to build a cancer vaccine? I think we all agree that we need an antigen, we need a good adjuvant, we need a good delivery system, we need a way to address the tumor microenvironment. And I think what's incredibly exciting about our current era is that we have this expanded toolkit that allows us to address each and every one of those challenges. We mentioned the checkpoint blockade antibodies. There are now clinically tested clinical grade adjuvants that are really quite effective. I won't go into it now. And there's different types of delivery systems that are quite nimble and flexible. I put long peptides as one example, but certainly there's a lot of interest in the field in RNA delivery systems, liposomal delivery systems, and so on. So I think the question becomes, well, what about the antigen? And I point to this paper that was published back in 2004. This is when I was an early faculty member. It was really like a major, major bummer. It was from Steve Rosenberg's group, and basically they pointed to the hundreds of cancer vaccine trials that had been performed, and with an objective response rate of 2.6%. It was really not a great time. And there are many myriad reasons why those hundreds of trials failed, but not the least being the selection of antigen. So again, I want to emphasize that it was a different time, there were different tools, but I think at least one aspect was the selection of antigen. So I think what was really not impressive was the fact that these trials tried to really go after a single antigen that would be the same for all patients. And I think I expended a lot of breath in the beginning of this talk trying to talk about how heterogeneous we are even from the genetic standpoint, not to mention all the other different aspects. So I think the place where there was some glimmers of success, some kind of little bright lights, at the end of the tunnel was these more complex vaccines. So whole tumor cell vaccines seemed to do a little bit better. And then there was some idea that maybe we could improve delivery, whether by long peptides or using dendritic cell-based vaccines. But in general, some idea of a more greater complexity. So a lot has changed. I mentioned already next generation sequencing and how it's availed us to much greater understanding of the genetic composition of our tumors. And I already mentioned to you the ability to kind of systematically look for neoantigens. So I think that's really, what's changed since the time that Rosenberg and Nick Restifo published that review. And what we really think is that based on all the evidence that I showed you earlier is that neoantigens may really be hitting the sweet spot because certainly they're exquisitely specific in terms of tumor expression. And akin to pathogens, there's a lack of central tolerance, and so highly immunogenic. And I really want to point out that this is really a different space than where we've been operating in the past, which is shared overexpressed tumor self-antigens that are variably specific in terms of their tumor expression and variably immunogenic as well. And I think already there's been nice animal data that have suggested that using a type of vaccination strategy that targets neoantigens is effective. So this is on the left work by Bob Schreiber, on the right work by Lilia Delamore and Genentech, using two different mouse models in which they targeted predicted neoantigens together with poly-IC, which is a TLR3 agonist, and then using a long peptide format, they were able to show good responses, protective, both in the prophylactic and therapeutic setting in mice. And then Steve Rosenberg has had a story where using TILs that were expanded against a single neoepitope that was personal for that individual. If you follow the bottom curve there, the first peak and downcline was just the total TILs, but then the second peak in the rapid down is infusion of the neoepitope specific T cells that were from that patient. So evidence of in vivo cytotoxicity. So with all this, can we actually target personal neoantigens as a multi-epitope vaccine? Well, so having built our pipeline and developed all these kind of ideas, we really set forth to try to ask this question, whether we could actually do this. And we thought long and hard about what would be the appropriate setting to do our first test forward. This was work done with Patrick Ott and many others, Ed Fritsch, Nir HaKohen, and a number of other people in my group, and really engaged both investigators at MGH, but also at the Broad and the Dana-Farber together. And the idea here was, as a first step, always thinking that it would be great to combine this with checkpoint, but we want to understand what the vaccine alone can do. To give us a little bit more wiggle room in terms of target selection, we ended up going with the tumor that has the highest mutation rate, melanoma. And we also wanted to be able to test whether or not there was going to be immune response against this type of vaccine. If we were to construct a vaccine that was against multiple epitopes that were specific for an individual, we wanted those patients to be able to go out long enough that we could actually test them. So this kind of led us to consider a adjuvant setting where there's no evidence of disease, and where there wouldn't be kind of also confusion with institution of other therapies. So suffice it to say, this was stage 3 melanoma, resectable stage 4. We were able to surgically collect a tumor, normal from peripheral blood. We performed DNA, RNA sequencing, HLA typing. We performed prediction of the HLA binding peptides. We decided to go with a format of synthetic long peptides, so not 8, 9, 10 MERS, but rather 20 to 30 MERS. There's a lot of preclinical data as well as clinical data to suggest that these kind of longer peptides allow, through cross-presentation, internalization of the epitope, processing, and generating more robust CD4 and CD8 responses that are more durable and long-lived. These peptides, we were targeting up to 20 epitopes. Again, the idea that we wanted to have a multivalent vaccine to avoid immunologic escape. These peptides were pooled into 4 different pools, so 3 to 5 peptides per vial, put together with poly-ICLC, and then injected as non-rotating injections at 4 different sites in the schedule shown below. So priming first to have high antigen exposure early on, and then 2 boosts later on. We've been able to kind of follow the immune response of these patients. So I mentioned before that a major question for us was, could we go from prediction to actually generation of immune response? And we made a big effort to collect blood at day 0 and also week 16. And across, at the present time, we've enrolled 12 patients. We've really spent a lot of time analyzing the first 6 patients. I didn't include the slide, but across all 6 patients, we see very strong responses that were not present before, rising with vaccination, that we can detect in the peripheral blood with just a short exposure over 24 hours against the peptides across multiple pools. And what I want to show here is that compared to week 16, we clearly see new responses against CD8 predicted epitopes arising across different patients. These T cell responses are polyfunctional, suggesting that they're really active, secreting multiple cytokines. And when we deconvolute per patient as to what are the personal epitopes against which we included in the peptide vaccine, we see responses against multiple different epitopes, both on the CD4 and the CD8 side, and that there's preferential reactivity against the mutant epitope that's shown in the red line compared to the corresponding wild type, which is shown in the black line. And what's been also very gratifying is that we've been able to generate melanoma cell lines from these patients, and when we look, just focus on the right-hand side, we can see CD4 T cells that can recognize autologous tumor, as well as CD8 neoepitope-specific T cells that can also recognize, in this other case, autologous melanoma and not irrelevant melanoma. So again, the concept that this is really quite a personalized product. So I think I just want to conclude by saying I think we're really, at least for cancer vaccines, really it's been a paradigm shift, because I think what's been really challenging about vaccines in the past is that there's so many different moving parts. It's hard to get one part straight when you're trying to juggle all the other parts. And I think what's exciting right now is we have a couple different anchor points. I think the neoantigens we think are a good class of antigen to go after. It allows us to anchor the antigen part, and then we can actually address the questions of schedule, delivery, and so on and so forth. And so just to summarize, I think there's been really many opportunities and riches that have been afforded to us by next generation sequencing, not the least being our ability to systematically now mine the genome for potential immunogenic antigens, and also to characterize the immune context. There's just several lines of evidence by now that suggest that tumor neoantigens are an important class to target. Tumor-specific responses can be generated. I've shown you just the example of one phase 1 trial that we're completing now in melanoma. We have a parallel effort in GBM that we've been conducting with David Reardon, which equally shows really quite interesting results. We have a new effort together with Tony Chuari in renal that is starting shortly, as well as some trials that are going to be initiated in the blood malignancies as well. And certainly we are very interested and hopeful about putting this together with checkpoint blockade, because I think really that's where I think will expand the classes of patients and tumors that can benefit from this approach. And this is the amazing group of people that I've had the privilege of working with. So thank you.
That was super, fantastic. I mean, this is, having been in this business for so many years, I can see how much more logical this is than some of our original ideas about making tumor vaccines, grinding the tumor up, injecting it, and hoping something happens. So, Kathy, good luck. And for all our cancer patients who are depending on this is something that can work. We have questions. We have a few minutes for questions. Somewhat familiar with the preclinical literature, which suggests that some antigens, specific antigens, are very potent in terms of initiating an anti-tumor response. Other peptides that may be presented well and be unique antigens still won't work as anti-tumor. So, anti-tumor antigens. So how do you distinguish between, is it going to be hit or miss, or is there something that tells you, is it all gamma interferon production, or is it just sort of, well, that's sort of the basic thing we're looking for, but we have to hope that what we found is actually going to work?
He always asks great questions. No, I mean, I think really that's at the crux. So I think what I tried to show you was kind of our thinking in our workflow, and getting from theory to actuality is a different, I mean, the more we dig in, the more we realize there's levels of complexity. So at the minimum, what you want to predict is something that's actually going to be on the tumor and that the T cells will see. So that's a lot to work through. And so what I would say is that we are systematically trying to work through those parts. I think the algorithms, the existing algorithms that we have only address the tip. And at some level, what I would say, it's amazing on our clinical trial that we even saw the responses that we did working with this kind of version 1.0 format of these algorithms. We are making a very systematic, dedicated effort right now to de novo relook at the prediction algorithms using mass spec as a way to discover the peptides that are displayed and retraining our algorithms. I think we actually need to do the same thing and understand how the tumor is processing these epitopes, how that is different than the antigen presenting cell. Are they the same or different? Because we got to make all these different parts talk to each other. So active work, not only by us, by many others.
I think the point is there are a lot of other things to learn about the immune response,
absolutely.
but you're doing it in a very logical way. I think that's beautiful. Okay.
Hi, Kathy. Interesting talk. I have one question. I think evolution is always getting better thinker than us. Like when it comes to chemotherapy, also you have a lot of normal mutations, and you target with one drug, and there are mutations which get selected and then expanded, and then you have recurrence. So how about this? Like now you have a tool that you can actually sequence what are the new antigens in the tumor. Mm-hmm. So tumor is heterogeneous. So you have like some clones which are dominated and some clones which are very few. So you make these synthetic peptides for the dominant clones, but in the meantime, you train the tumor to have less heterogeneity by just grinding up the tumor and giving to the system, so that all the small clones will be eliminated and the tumor will be less heterogeneous now. And then you come with your synthetic peptide specific CAR T cells or whatever vaccines you are making, and then you can eliminate all the heterogeneity, right?
Yeah. So I think, yes, I think we're trying to treat, also knowing that there's kind of a moving train there at the same time. I think one of the things I like about our personal vaccine approach is that it can be reformulated at different times. So as we get better and better at what we do, I think the time scales for resampling, reprofiling, regenerating vaccines, reformulating the content of what those vaccines are will be possible. I think kind of the other concept that I want to bring out is that as much as I would like vaccines, a multivalent vaccine that will hit many different targets to avoid this kind of escape. I mean, we have examples in microbial immunity and so on and so forth. I mean, I think we need to hit from many different angles and corners to deal with the cancer mass. But as much as I would like the vaccines to be as potent as possible, I think it would be a lot of hubris to think that the vaccine by itself will do everything. And so I think there is a very strong role for combining with effective cytolytics that are not toxic to really bring down the tumor mass, and then to combine together with vaccine, combine together with CAR or checkpoint. So I think it would be cancer is a hard problem. So it's hard to imagine that just one modality is going to do everything.
So what do you think is a better method to decrease the tumor heterogeneity? Like,
That's a tough
Yeah, I think that is a tough question, because I think first of all, there's lots of different cancers, and I think in terms of, at least, so I can speak about CLL, and what I see in CLL when we compare chemotherapy versus targeted therapy A or B or C, I mean, it's different depending on each therapy, and I think we're going to have different answers depending on the tumor and the therapy.